there are many virus structures are there in between 2 to 3 angstrom. So you can easily reach that because virus particle itself, it is a very big molecule, as well as the molecules are symmetric. The icosahedral symmetry is there. That is one of the reasons your resolution will be very high.
And if we want to study like some molecular processes going on inside the virus, like it is infecting the some cell, and we want to study the like mechanism of its how it is entering into the cell and doing its function. So can we use the cryo-electron microscopy to study this kind of functions, like its entry and also mechanism of action to some extent? Can we study like?
Yes. So you have to understand one particular thing that, okay, if you just look at my hand and if you just consider that, okay, there is a movement of that finger, or if you just consider that there is a watch. So this is the mechanism that I'm tying up this watch, right? So now if we want to continuously see these things, how I am doing this entire process, in the microscope, you cannot see that entire thing that I'm taking it out, I'm putting this, and then I'm putting these things back. So that is how I am wearing this watch, right? So that entire thing, because this is the live activity, I am doing it. So live activity you cannot capture, because in electron microscope, everything will be under high vacuum. And high vacuum, nothing can be live when there is a high vacuum, oxygen and other things are not there. It will be not living object, right? And the another part is that what you are doing, you are taking a snapshot. So if you can trap those snapshots that, okay, there is a viral infection, if there is a cell culture and there is a viral infection. And then one case that this is the cell, virus will go and just bind. And another case, virus is already interacted and the DNA is going. So you have to search that entire area. You can see that snapshot, but you cannot see that live cell imaging kind of thing. That you have to understand. Mechanism, definitely you can see it, how it is interacting with the cell, how DNA is going inside, how it is processing. All these things you can capture, but that will be a huge world. You have to spend a lot of time to visualize one by one this snapshot, and then you have to connect all the dots, and then you will get the entire picture.
Okay.
But the continuous process is happening. What I was showing with this wristwatch, that how I am wearing this wristwatch, that cannot be visualized by Cryo-EM or TEM, because that is not a continuous process capturing equipment.
Okay. Thank you.
Okay.
Yeah.
Thank you for this question.
Yeah.
And how is the overall course? You like it? Is it okay?
Yeah, it is very fruitful for me because it covers almost everything in the cryo-electron microscopy. So right from the basics and to the little advanced level also. So it is very beneficial to me. So thank you very much for taking this course, and yeah, thank you.
Okay.
Yeah.
Okay. Next question.
Hi.
Hi.
Hello, Professor.
Yes. Hello. Karthik, right?
Yeah.
Yeah.
So I was wondering, I was a little confused about how do you produce a parallel beam of illumination?
How do I produce the parallel beam of illumination?
Yeah, that hits the sample.
Hit the sample, but how I can explain it here, sir? That is the problem, because in the laptop, I am not in that conference room where I was recording all these things. So just look at this one. Can you see my board? Can you see my board?
No, I think you've blurred the background, so it's some distance away, and so it can't be seen.
How to unblur the black background? Anyone can help me little bit from NPTEL one? Then I can unblur and I can show him how to do it. That is the reason I am at the classroom. So I can
1 second, sir.
say again.
1 second, sir. Hold on.
Yeah, yeah. Whenever you are ready, you just let me know so I can start the drawing.
I think the way to do it is to, so when you go to the Zoom app, like there is a settings button, like one of the tabs.
Setting tab.
It may be like near the top of the screen.
That is I'm trying. That is I'm trying. No, so from the NPTEL, someone told me, wait for a second. I thought they are probably helping me. That background thing, I know how to do it in Teams, but not here basically.
Okay, so can you see the place where you can see like about Zoom? So there is one tab that shows you about Zoom.
So I can see view. This is showing speaker, gallery, multi-speaker.
No, Professor, like above that, like within, your computer.
Hold on. They help me. They help me to do that. Okay. They bought that screen for me. So you did it? Okay. So let me do it then. So what is happening here, if you remember that microscope, what do you have? Why it is so? I don't know. Okay, let me do it in my own way. New slide. See my PPT?
Yes.
You can see it, right?
Yeah.
Okay. Okay, fine. So if you remember that how I was teaching that initially you have that C1 lens, right? This is your C1, then it is coming C2, and some cases you have a C3 lens also. If you don't have the C3, what you have the next, you have C1 lens, then you have the C2 lens, and then you have the objective lens, which is called the twin lens. That objective 1 and objective 2, objective 1 and the objective 2 lens, right? So objective 2 lens, this is called the twin lens because this lens are splitted in something like this one. Now, here they are interconnected, these two lenses, and how it is coming, if we just start from the beginning that, okay, gun crossover point, it is there. That is the D0 I talked multiple times. So first beam, it is coming like that. This is your C1 lens current. Then C1 lens current, it is converge the beam somewhere here. So now this is your C1 lens. When you are considering the C1 lens, this is your spot size we are considering. Then after C1 lens, what you have the next? You have the next that C2 lens. C2 lens basically your intensity lens. Now, if I have the C2 lens, it is going, and then you can get a crossover point somewhere here. Crossover point somewhere here. Now, your objective lens is here, these two lens. If your crossover point is here, you are keeping a magnetic field in such a way that you can make the beam like this fashion, okay? and it will go like this way through your sample. So what you are doing basically that, okay, you have a crossover point here, and then you are trying to make whatever electrons are coming, you are trying to make the electron beam coming in this fashion from your crossover point. And that crossover point will be this one, the second crossover point. Basically, we are trying to make this one parallel like that. Another way to make the beam parallel is something like that. You have a first crossover point here. This is the first crossover point. Then your C1 lens current, and then your C2 lens current, you make it in such a way that your entire beam, it will go like this fashion. So there is a two-way to do it. So in this particular case, you are making your lens so under focus that C2 lens current is making so under focus in this particular case that your beam is looking that semi-parallel kind of thing. And other way is that you will make a beam cross over here and the magnetic field in such a way that it will take only a small place, that this is the small crossover point, and all the electron, whatever it will go in a straight fashion. It cannot deviate in any direction. This is how you will make the beam parallel.
Okay,
Is it
so
clear, Karthik?
Just recapping, so the role of the C1 lens is basically to use a high what's it called, high lens, use a strong magnetic field basically, so that the beam the spread of the beam is large, so that a lot of the electrons will not go through to the C2 lens. Like for imaging biological specimens, we need to keep the dose low, right?
Oh, that is a completely different thing, because whatever first question you asked me, sir, how you are making the beam parallel, that is I am telling that there's a two-way to make the beam parallel. Okay. One is using the C2 lens under focused. So when C2 lens is under focus, this is a parallel beam. Otherwise, C2 lens, you will make a focus just above the sample, and then your upper objective lens current, you will make it in such a way that from that spot, if this is the spot, all the electron, whatever it will come out straight like that. Okay. Then it will make your beam parallel and it will not expose. In this particular case, it will expose the entire grid. But in this particular case, it will not expose the entire grid. It will be a small part, it will be exposed. Now, your second question is that C1 lens. C1 lens function is completely different. C1 lens is called the spot size, right? So if you are using a very strong magnetic field, you can see the strong magnetic field means your crossover point is going very up. It is going in up this direction. And now you can see your beam is spreaded like this one, right? Now, if your beam is spreaded like that, and if you put a aperture here, you are going to block your most of the electron beam. So that is one of the reason for biological science sample, we are using a strong C1 lens current or magnetic field, which will change the crossover point towards up. Beam will be spreaded too much, and then use the aperture and block most of the electron beam.
Okay, and what does the C2 lens do for imaging biologicals, like
C2
low
lens
dose?
is basically your intensity, and the beam diameter changing, it is regulating. So C2 lens basically, it is helping. If you look at this one, C2 lens is helping where your crossover point will be. If your crossover point, your electron beam will go like this, or your crossover point will be somewhere here, and then you are going to make your electron beam parallel, or you are just want to make your electron beam something like this. So C1 lens basically helping you to both C1 and C2 lens work together with the aperture and decrease the dose. And C1, C2 lens basically helping you where your electron beam will converge. Is it just above the sample? It is on the sample? It is below the sample? Or little bit far away from the sample? That is basically controlled by your C2 lens.
Okay, and so we keep adjusting the strength. And so when we do low dose imaging, we want a strong C1 lens and a weak we want to apply weak field strength on the C2 lens.
Yes.
Okay.
Okay.
Okay. Thank you,
Did
Professor.
I answer your question?
Yeah.
Okay, thank you very much. Any other question? How do you like the course?
Yeah, I enjoyed the course.
Okay, thank you very much.
Yeah.
Who else?
So I have a general question, not related to it is related to the course, but it's very general. Means we can have these videos which you have uploaded. When we can have these videos until what time?
That now you have to talk to NPTEL office people because I am not uploading anything. I
Okay.
just uploaded the course. I gave you the assignment. I gave you the question paper and I'm discussing and answering your question. But rest of the official stuff that, okay, how long it will be available, how you can get it, only the NPTEL office can answer these things.
Mhm.
I'm not taking care of those part.
Okay. Okay. So, sir, is any free software which we can use for data processing of the we have some data collected from any Cryo-Electron microscope source, and we can have any free software we can.
So all these softwares are free so far.
Okay.
So if
Okay.
you just consider E-man, Reliance system, for
Mm-hmm.
the academic users, all these softwares are free.
Mm-hmm.
Most of the cases you need a Linux system. In
Mm-hmm.
that Linux system, you have to install, you have to know little bit details about how to run that. And time to time, we are organizing many workshop for the data processing, model building related stuff in ISC and NCBS. And generally we advertise whoever is interested, they can come and they can learn. Just 2 months ago, we did a similar kind of thing, that hands-on training about Cryo-EM, TEM, some data processing workshop. So
Okay.
many students came from all over India. So we are doing these things regularly because those data processing tool probably you have to learn little bit more
Mm-hmm.
because that will be not a very straightforward way to do it.
Mm-hmm.
A little bit complicated, but it is doable. And I'm also thinking to take another course about how to do the data processing.
Okay. So that would be very near, or means would you advertise for that very soon, or it will?
Yeah, maybe next year.
Okay, next year. Okay. Okay. Thank you.
Okay.
Mhm. Thank you.
No more question. So if anyone is here from NPTEL office,
Yes, sir.
yeah, so it is advertised to everyone, right? All the students, because I cannot see many students. Very few are here basically.
Yes, sir, it is advertised.
Okay, okay. So I'll just switch off my video if someone, do I have to wait until 1:45?
We'll wait
Yeah,
for 10 more minutes, sir.
yeah. So I'll just switch off my videos.
Okay, okay, sir.
I have another question.
Thank you very much for your
Yeah.
question. Basically, I was sitting alone here, and if you guys talk, I'll feel very good.
Okay, sir. So I was just wondering, so you had mentioned that basically these lenses, so since we are using this electromagnetic lens, which basically has this copper coil windings, and you basically pass current through them to change the field, they can suffer from hysteresis. So practically, how does that affect imaging? Like, what do we have to keep in mind, and what, like aberrations or something we can see because of this hysteresis effect?
Okay, hysteresis effect, if you remember that I told you, okay, let me share my PPT again. That will be easier. Share it and share. Okay, you can see my PPT, right? Here.
Yeah.
So in that PPT, if you recall that, what I did it previously, the curve was like that. And then one way this curve is going like this one. So if you just consider that there is a metal bar, that iron or nickel or cobalt. So there will be a south pole and north pole, right? And we are always using the ferromagnet for making that soft iron for making those kind of lens system. Now, in that ferromagnet, there is a single atom and it has a tendency for north and south both. And now, based on this electromagnetic field also, if there is a magnetic field it is generating, that most of the time this north and south, if you just consider, there will be the magnetic field, it will go like that, right? So even for the gravitational force also, you can see similar kind of thing. So here, when there is a north and south pole, there is a division and it is going in one particular direction. So when those things it will align, it has some kind of that metal, when it will magnetize, it will follow certain path, and that will go in this particular path. And when it will come back to a certain to a normal one, it will follow a different path. Both the cases, path will be not same.
Because the
Now,
magnetic domains have to like reorient.
yeah, reorient. And then you have to give some extra force to put it back into the zero position. Now, in this particular case, what is happening, that what you have, that when you have a very low mag means that you have a very low mag means your magnetic field is not that strong. Because if you just think that, okay, you want to image this particular dot, right? So the electron beam is coming like that, then they will converge some particular point here, and then it will give you an image here. So here you can see the magnetic field is not very strong, it is weak. Now, if I have a very strong magnetic field, when I have a very strong magnetic field, it will spread. It will spread all over like that, and it will give you a very large image. So what you are changing the magnetic field. So what I told that time, sometimes you have to go to very low mag, and then you have to go to very high mag, because if you are in a very high mag, it will be very difficult to find out what you are looking for. So you have to be a very low mag. If you recall that I told you that you have a hole size. So hole size generally at 380x magnification, you have to check it the hole size. Then you have to go to little bit larger mag, that is will be 4K mag. And that 4K mag, you can see the hole properly, and in that hole where you want to image, you just consider this box we are going to image. And when you are going at 53K, now, if you just repeatedly do this, that okay, you are imaging something at 53K, you cannot see the entire hole. You will just see this small area. That small area, you can see it in 53K. Right? Now, if you just constantly do this, you are going to that very low mag here, and then jumping around here, collecting again. So back and forth, low and high. And you are doing it very quickly. Then what is happening? Your one crossover point is here. Your magnetic field is very low. Suddenly your magnetic field is very strong. Again, you are going down. So these changes, you cannot follow the same pathway. That ferromagnet cannot behave in a same way. So then what is happening? That is the lens hysteresis. Now that thing will be embedded there. And now when you are thinking that, okay, I am using a certain magnetic field or certain defocus value to image one thing very perfectly. But already your magnetic, because of your bad handling, magnetic field, it is not following the same path. That the metal is already, some magnetic portion is already stored. So whenever you are
Okay.
certain magnetic field for a particular magnification, it will add more. And then it will give you a wrong magnification, wrong defocus value. That is basically lens hysteresis and contributing to your image. So image, whenever we are considering that we want to image a very perfect image, very high resolution, 2 angstrom or 3 angstrom, that is going to affect for this lens hysteresis, because we are not following the protocol that whatever magnetic field we are considering for the defocus or the magnification, it is not exactly same value. It is changing because of this lens hysteresis. Is it clear?
Okay, and how do we like what steps should we follow to ensure that even if we have to go from lower to high, this hysteresis doesn't come into the picture? Like
It will be better to give some time. Okay,
how
that's
long?
the so generally it is very difficult to tell exactly how long. This depends on your depends on your resolution also. Lot of time for negative stain microscope, people are going very low defocus and then suddenly going to high defocus and collecting the data, because they just want to see couple of images. They are not considering that they can get a 2 angstrom map from a negative staining 10 images. And so, but for the cryo microscope, even that is the automatic things, it is coming. Whenever you have a low magnification, you are going to high magnification, automatically they are giving to 15 to 18 minutes to wait time.
So it's set in the
So
software.
that will be a error message. It will come automatically. It will be not an error message. It is a warning message that you change from very low mag to very high mag, and because of the lens hysteresis, you have to wait certain time. And then it will automatically stop the imaging.
Okay, and how do we know if hysteresis is taking place? Like if it's affecting the current image during the run?
So current image during the run, you have to look at your images. So sometimes images you can see some kind of pattern or some kind of deformation will be there. Today, I don't have any tame images with me, so I can share today. Maybe next class, I'll try to show a couple of images, how the difference you can see it between the lens hysteresis and without hysteresis. And also if you try to process the data, you can see that signal is significantly affecting or CTF is affected. From those things, you can get to know that the images is affected, but it cannot tell you particularly is it coming from the drift or is it coming from the lens hysteresis or is that there are some environmental issues. It will be very difficult to tell it from the image that it is particularly coming from.
Okay, so like the microscope cannot monitor the actual field present in the electromagnet.
At least whatever microscope we are using for the biological 10 or Cryo-EM, we cannot, but some modern microscope maybe, that I am not very sure.
Okay. Okay, and last thing is so you said we can wait, but are there any other steps maybe within the microscope's design or like in the way we do it that we can take that can minimize this effect, like other besides waiting?
No, so look, that is every instrument has certain protocols, right? So whenever you are inserting your grades or sample, then if that is a problem, that okay, you are using soft iron or soft metal that is making your electromagnetic length, and if you have the hard metal, that will be, it will take little bit longer time, or you need external force to bring it down to the zero. So it is always better whenever you are inserting the sample, you should not jump from very low mag to very high mag. So you just do these things gradually. So it is when I'm talking about electromagnetic lens, you just have a button, you can rotate it easily, and that is why from low mag to high mag you can easily jump around. If you just consider the glass lens, it is always taking some time, right, to take out the one glass lens, put it the next one, and then stabilize, then you are imaging. So same thing you do it here.
Okay.
There is nothing that everything you have to do it in a microsecond time frame or nanosecond time frame, right? So you inserted the grip, now you wait, the thing should be stabilized, the mechanical drift, it will be less. Then you have a low magnification, you selected some area, then go to high mag, then medium mag, from low mag to medium mag, then SA mode. When you are SA mode, you go to particular one magnification and then do it. So there is nothing that we have to do everything in a very fast mode.
Okay.
And if you follow that one, most of the time that lens hysteresis problem will be less.
Okay. Okay. Thank you, Professor.
Huh?
Thank you.
Okay. Thank you for your question.
Yeah.
So you are from which university? Just I'm curious, that's why I'm asking.
I've graduated, but we've met before.
Okay, okay. Okay, I'll stop sharing again. So I think one new student joined. If he or she has any question, we can. Any more question?
Okay, I have another one.
Okay, I'm here.
Okay, so I think this was in week 7. So you discussed the carbon sandwich technique.
Carbon sandwich technique. Yes, I described.
Okay, wait, is it carbon sandwich? So the one in which basically, I think I might be confusing it, but basically the what's it called? Like this, I think a thin like metal is vaporized and then it's deposited on the sample, and the instrument is kept like at an oblique angle.
Can you share me exactly which?
Okay, that maybe is it called metal shadowing?
No, metal sandwich is completely different and carbon sandwich is completely different.
Okay.
So metal shadow mostly we are using to see the DNA. Okay, that
Uh-huh.
is I explained that what you have the carbon coated grid, and then you have the DNA. Okay, and then you need the platinum wire, and then that platinum wire, if you just consider that, okay, this is the rod, and then you just rotate the platinum wire like that. Okay, and then there will be another carbon rod will be attached like that, and then when there is a heat, you will increase the current between these two carbon rod, it will generate some kind of heat, and the platinum will start evaporating.
Melting,
And yeah,
evaporating.
melting means it will be small atom kind of thing, it will start evaporating. Now you just consider that this is my carbon, and this is the DNA, right?
Mm-hmm.
This is the DNA. So now if you just consider DNA is spreaded here, and then one of the side, because that entire system, that entire platinum related things, it will start rotating. So evaporation, it will be uniform. So, and it will spread. And then one side of the DNA, constantly metal will deposit.
Mm-hmm.
And when the one side of the DNA of the metal, it will deposit, it will be thick metal, you can see it on top of the DNA. And that is basically metal shadow technique. And carbon sandwich technique, what I told you, lot of time previously, scientists are using carbon sandwich, and it is a very useful technique. Even if
Mm-hmm.
you just consider long back, when we don't have the cryo freezing system, people are using the carbon sandwich technique. What is happening? You have the carbon coated grid,
Mm-hmm.
right? Now you add sample. Then you have some kind of sucrose or glycerol kind of thing that will put it on the sample. So you have the sample, you do the negative staining on top of that sample. Then you have some kind of sucrose molecule or the glycerol, and then there is another carbon layer, you put it here. So there will be a two carbon layer 1 and carbon layer 2, and in between sample it will be there. And in between sample, there will be glycerol or sucrose molecule will be there. So it
Mm-hmm.
will not be flattened completely. So most of the time, negative staining sample problem is that samples are flattened, sample are collapsed. But
Yeah.
here, because of this sandwich, in between that glycerol or sucrose will be there, and then protein molecule will be there. That collapse problem, it will be not there. And even if you try negative staining, you can get much better actual the morphology of the protein sample, mainly the virus sample.
Okay. Okay.
So
So,
these two
mhm.
techniques are different. One is carbon sandwich technique, another one is that metal shadowing technique.
Okay.
Okay.
Yeah. So I had a question about this, actually about both of them, but let's first see the metal shadowing one. So so I got it that basically you have like you keep this the wire of platinum or whatever other metal you want to use at an oblique angle to the surface. So it's like one a small acute angle, I guess you could say, from like the
Can you
horizontal.
switch on the video, and can you tell me what you are trying to say? Then it will be easier for me to,
I'm sorry, I can't right now.
okay, okay.
No, I'm just saying that like suppose the sample is we assume the sample is the horizontal level, the where the grid is kept. So if we above it the wire is placed, but the wire is not exactly parallel to the surface. It's at a small acute angle to the surface, right?
Yeah.
Yeah. So and because of that, it the metal tends to like deposit on one side, like the side
Yeah,
that is
one
closer
side.
to it.
Yeah, because always it will be not a very flat sample. That is exactly I was trying to
Mm-hmm.
show that if this is the grid and if this is your DNA, what will happen one side, because it is not the exactly flat surface
Yeah.
anymore. So one side it will be deposited. I think maybe your my video is not that clear. So here you can see this is one side, that is the other side. So
Yeah.
when there is a metal will just deposit in one particular side, not both the side.
Okay.
Okay.
Uh, yeah, I was just wondering, so how exactly does this produce better contrast? Like, how is it that because one side is shadowed and the other is not, we are able to see the DNA, and negative staining cannot give us that same result?
Okay, so if you just consider that normal carbon coated grid, right?
Mm-hmm.
Normal carbon coated grid and DNA is a very thin sample. So now if you
Yeah.
just consider the carbon coated amorphous carbon and then DNA composition, it is very thin. So it is not like a protein which has a globular structure and multiple helix is giving some kind of thickness. DNA means it is just a thread,
Mm-hmm.
very thin thread. Now, that is one of the reason it is very difficult to distinguish your carbon and the DNA. And
Okay.
that is the reason when there is a metal shadow is going through your set. So it is something like that. So for the course purposes in the ground, you can just use a stick and draw all the track.
Mm-hmm.
But the runner cannot see that one, right? That is the reason we are putting some white boundaries on the ground. So definitely when you are marking with a stick in the ground, that mark will be there, entire
Mm-hmm.
100-meter race. But can the runner see it? So runner cannot see it. That is the reason we are putting that, what is called. So
Mm-hmm.
that white powder we are putting it all over so we can see that track properly. Exactly same thing we are doing because DNA cannot be distinguished from the background of the carbon. So we are just using the metal. So one side of the metal, it will shadow. So that will be metal means it is a very dense molecule. Whenever electron beam, it will go through that, scattering will be stronger. From the carbon, you can easily recognize what is your DNA.
Okay. And like in a negative stain, like it's like the solution covers the entire area. So over here, it's more focused on like the
Yes.
contours of the like the shape of the DNA. It will
Yeah.
pile up near the top and okay.
Yeah, so I can add one more thing here.
Mm-hmm.
Definitely, the metal shadowing is the one of the best technique because previously those images very few people can do it in recent days also. So
Okay.
whatever image or references I showed in the class, but same time I want to add even I imaged many DNA protein sample without metal shadowing. It is just a normal uranyl acetate staining. So you can see nowadays even if you optimize the negative staining technique, you can see DNA on the carbon
Okay.
coated grid also. But you have to be some proper protocol you have to follow to stain the DNA and visualize in carbon coated grid in
Okay.
normal negative staining procedure.
Mm-hmm. Okay. Does somebody else have another question or should I ask my second question?
It is not looking anybody else wants to ask because I saw one new student join. Funny, but no, no response from the other end.
Okay. So my second question is, so I read online that there are a few reasons why the carbon sandwich technique it tends to produce better data, like better micrographs. One of them was apparently that there is some symmetry between the layers. So I think there is some beam induced motion, I think, even in the when there is carbon sandwich technique, but somehow because there are two carbon layers, it somehow cancels out. I did not understand that. Could you please, and what are some of the other reasons why this technique is better? So you've already said that because glycerol is present, like the
So
sample is a little more hydrated and it's not completely
yes,
collapsed.
yes, exactly. Hydrated. So now I don't know where you rate that one, and because if you don't, you give me the reference is bit difficult because so different when you are looking it online or any manuscript. So sometime that particular manuscript for particular sample, that particular situation, that was the best. But if you look at the current scenario, that carbon sandwich technique, no one is using carbon sandwich technique. Our cryo techniques are so much evolved and so much giving the better resolution and better data. So we don't use the carbon sandwich technique very frequently. So now, are you comparing with the normal negative staining data with your carbon sandwich data, or what is the comparison? What is the baseline here? What is the reference based on that you are comparing that carbon sandwich technique is better? Definitely, carbon sandwich technique is better because sample will be hydrated and there will be that if the sample is completely not in ice, amorphous ice, there will be a stable carbon platform. The beam induced motion will be very low. So if you just consider, if you have a hole, this is the hole, and your sample is in the ice, and then you are putting the electron beam there, definitely your ice will be, the ice thickness may change, it will be bulged. There will be some other changes in the ice, because water is always tend to stay in liquid or gaseous form. But if you have a carbon layer, there is a base, right? It will be not moving like a water molecule or amorphous ice.
So the beam will not
that-
pass but I was thinking the beam has to pass through the full construct anyway. Like it has to go through the first carbon layer, through the, like the aqueous slash protein
Yes,
layer, and then so like how does it matter so much that there is a carbon layer on top for the motion?
carbon layer on the top at the motion. So look, I don't know which reference, what paper you are using. So if you can share me the reference, it will be
I'll have
better.
to look.
Yeah, better, because nowadays online there are many things, many places are there. That is the exact reason I told you, for even for a particular manuscript. So even they try to do something, and they will tell that okay, their technique is the best technique. This is the reason. But why it is best? That I also have to check what reference they are using. Based on that, they want to
Okay.
comment. So I don't want to comment anything until I'll see that manuscript. Okay.
Okay. But
Yeah.
like for the general technique, could you tell me something? So like the general technique also involves two layers, right? Like the standard one that you talked about in your PPT.
No.
It does not. Okay,
No.
so
So whatever technique I discuss mostly, I discuss that normal negative staining, that is a one single carbon layer, and I discuss about the cryo. So I didn't discuss too much about the carbon sandwich. I just talked little bit about the carbon sandwich, how previously people are using. But
okay,
whatever cryo I discussed, that cryo thing, mostly it is a single layer. There is no double layer.
okay,
Okay. And also I probably I discuss little bit when I talk about cryo-negative staining. The cryo-negative staining also there is a carbon sandwich technique people are using.
okay. You can do cryo-negative staining.
Cryo-negative staining, but those things in 2009, 10, 11, it was very popular. But nowadays, how the cryo microscope is behaving, most of the time we are not using those techniques anymore.
Okay. I was just wondering if it can provide more useful information about the quality of your sample than just plain negative stain.
Say
Like
again, repeat
so
again.
when you're optimizing your sample, preparing for your initial cryo-EM run, would it be better to, instead of just doing negative stain, maybe do cryo-negative staining to see what is the quality of your sample, the monodispersity, and
No, I think negative staining will be good enough for checking the samples are monodisperse or not, and sample quality, concentration, everything. So you don't need to go to cryo-negative staining, because whatever information you can get it, you can just directly go for cryo. There are many labs are there, they don't even do the negative staining. They just take the sample and put it directly in the cryo. If you just add the cryo-negative staining, that itself, it will be little bit costly, not little bit more costly. Always remember that cryo grids are expensive than normal TEM grid. Okay,
okay,
it's almost 10 times more. And then when you have to insert those grids, you need the cyclic ring, base ring, that also cost more. And then microscope timing. And
okay,
also you have to remember that you are using uranyl acetate or uranyl format that may contaminate your microscope and your freezing instrument. So there
okay,
are so many hurdles are there. So better not to do that if it is absolutely required.
okay. I guess a follow-up question is, so why are the cryo grids more expensive than the normal ones?
Because you need a uniform sizes hole. Okay, so
Mm-hmm.
in the carbon coated, if you went through my, did you see all the videos properly? Because in the video I discuss about all of those things. Because
Uh-huh.
that is the basic difference of the grid. The carbon coated grid, there is a continuous carbon layer, and the quantifoil, there is a uniform sizes hole. And when you have a amorphous carbon layer and you have to make a uniform sizes hole, the making entire preparation technique is different. And that is one of the reason it is, costly.
Okay.
It is
Okay.
time consuming, more reagents are required, more expert people are required to make that one. That is one of the reason it is expensive.
Okay.
Any more question?
I guess that's it from my side.
Okay.
Thank you again.
Hello? Anyone from NPTEL office?
Nothing, sir. I think we can close the session, sir.
We can close the session, right? No one is asking any question, anything?
Yes, sir. Yes, sir.
Okay, so we can stop.
Yeah, yes, sir.
Okay, thank you all for asking question. Thank you.
Thank you.
Thank you, sir.
Yeah, bye.